Perfect.
All right. Hello, everybody. My name is Robert Sam. I am a student at the United Tribes Technical College, and today I will be talking to you guys about the project I've been working on. So the project I was working on was characterizing vegetation using neon remote sensing data, and we were looking at structural diversity metrics. My mentor was Chelsea, and my technical mentor was Leah, but I had help from everybody during this project. So if you have not heard or do not know what NEON is, NEON stands for the National Ecological Observatory Network. And um, NEON provides open access to data that they collect at their many sites across the United States. And they make that data freely available. And of that data, we are focused on the airborne remote sensing surveys. And so these are collected by some instruments that they use, and they are collected during a peak greenness of their testing sites. They occur every 3 to 5 years, and one of the data products that it produces is LiDAR data, and that's the one that we are focused on the most. So LiDAR data is collected both discretely and in waveform. Some of the data products that come out of this LiDAR data are discrete return LiDAR point cloud, LiDAR slant range waveform, slope and aspects, elevation, and ecosystem structure. And that is the one that we also wanted to focus on for this project. So we looked at ecosystem structure and diversity, and we were looking at structural diversity among forests. And we were led to this by a previous research that was done for the eastern side of the United States, where they calculated structural diversity metrics for various different forests, and they were mostly on the East Coast, and so we wanted to do something more on the western side of the United States. And so that led us to the question, how does structural diversity vary across different forest ecosystems? So we started out by creating our workflow diagram, and this gave a very clear pathway on where we should start and how we should end and how everything should go according to plan. And so from our workflow diagram, we then selected the forests that have data available from NEON. So the forests that we choose were in Washington, and this would be the Pacific Northwest, and that was called the Wind River Experimental Forest. And then the soap root saddle, which is in California. It's on the West Coast, around the Sierra Mountain Range, and it's called the soap root saddle. So from there, we downloaded the data from NEON and began coding in RStudio. So I started with the soap first, and I plotted to see what I am working with, and that plot is this one right here. And so this is the LiDAR data. When you plot it, you can move it around and basically choose where you want to study from. So from afar, it really just looks like a plateau, and I'm trying to do this as best as I can with my mouse pad. But if you scroll in just a tiny bit, you can start seeing more detail. As you can see over here, we're going to have trees, and it's a little bit spread out, and you can see some shrubs in the mix too, and a lot of bare earth right there. And so from there, we went to size it down and pick a 200 by 200 meter sample size, and that was this one. So we plotted that again. We have it down to what we want. And we were going to go again to a 40 by 40 meter, but that just made it hard to visualize and see what we were exactly looking at and working with. And so we decided to stick with the 40 by 40, and you could clearly tell what you're looking at from the top and from the side. So now, yeah, so now we started calculating the structural diversity metrics, and we then turned those 13 diversity metrics into a function in R and applied it towards the Wind River dataset. And then from there, we combined into the table. But I just want to show you what the Wind River looks like. And this one's a little bit hard to pick out and see, but as you can tell, it is heavier dense with trees, whereas the other one was very spaced out. So here's one of the plots, and then we did the same thing with breaking it down to 200 meters, or a 200 by 200 meter square, as you can see there. And then again, a 40, this one you could somewhat see a little bit better, but it still is very difficult. So we decided just to stick with the 200 meter one. And so once we did the 13 structural diversity metrics, we combined them into a single table. And you can see that the Wind River experimental forest has a higher mean max of a canopy height, where they're 38, and everything is in meters. I should have specified that earlier. But they're almost twice as tall as the soap root ones for the mean max. And some of the definitions in there that might not be as common or you might not understand are rumple. And rumple is the ratio of surface area of the outer canopy to the surface area of the ground. And this just kind of gives you a visual, and it helps you understand these two forests compared side by side to each other. And so here are another two plots that we did, and you can see them from the top. The soap root is really spread out, whereas the Wind River is kind of all clumped there, and you just have these certain areas where there's no points, or it's most likely bare earth. And so the two forests showed structural diversity from each other, and being that they're both on the western side of the United States, they're in different climates, which has an effect on both species and structural diversity for these two. But I would like to compare these two to others that are in the same region, or maybe compare it to the project that we used as a guideline and a reference. So I want to thank everybody at Earth Data Science Course and United Tribes Technical College, and my mentors and peers for everything throughout this whole entire internship. Thank you.